While you're thinking of questions, I guess, Bill, I didn't set you up, but here are the examples, right? Collaborative scientists, humanized models that you discussed as being essential. And are we done? Or the serious question is, I've read about, and you probably know more about, the concept of what has been talked about as the Center for Cures at the NIH. And are there models that can be applied that get us there faster, cheaper?
Yeah, I don't know much about the Center for Cures myself, but I think these type of presentations, let me just talk for a while.
Well, give us this. Okay.
We can speak up. Yes, we can speak up. The type of things that you see here are what happen every day in an academic health center. It's the stock in trade of what we do. And I think that's, to me, the excitement about an academic center. When I talk about academic centers being the translational engines of the future, I'm talking about this type of work, where PhDs and MDs and basic scientists, physicians, and clinical physicians are working together with disease models and the tools. And we've seen just two excellent examples of it. I mean, I think it's a great juxtaposition of what can happen within an academic center. Were I investing at a policy level, I would invest heavily in this, actually, within academic centers, and use things which drive people into these type of reactions rather than the funding agencies would typically drive them apart. And I think that's part of the transition we have to make in our thinking here. And these team approaches that use multidisciplinary teams have to be incented, rewarded, nourished in many ways. Some of them endogenous within medical schools, promotion space, etc., and some of them exogenous by funding agencies. You know, ultimately, most places and academic centers go towards nutrients and away from regulation. And in fact, we migrate just like giant amoebas to that. And in fact, the incentivization is the funding and recognition, and the idea of cutting out wasted time and space and regulation is really the kinds of things we drive away from. And I think that's what's the milieu that can happen with the right leadership within an academic center. So I think these are great examples, two very exciting projects.
Thank you, Bill. Alan?
First, I want to thank each of the speakers. These are each terrific presentations. I wanted to just pose a question contrasting Teresa and Vern's work as it relates to the T cells. Teresa is showing an example of targeting a very, very specific population of T cells. And in Vern's case, the T cell leukemia is intuitively obvious how you want to just obliterate all of it. But what are the thoughts about T cell depletion? I know people have used anti-T cell antibodies in the depleting approach, but in diseases like psoriasis or type 1 diabetes, is there any glimmer yet of what the implications will be of sort of wholesale depletion of T cells if that is what occurs with the inhibitor?
Well, it turns out that only dividing T cells are affected by this therapy because only they have the gene expression profile that transports and phosphorylates the deoxyguanosine to deoxy-GTP. So if we take quiescent T cells from volunteers in our lab and treat them with these drugs, nothing happens. If you stimulate them with high concentrations of IL-2 or foreign antigens so that they're dividing maximally, then they become highly sensitive. So it's only the rapidly dividing ones, the what I call the National Guard, or what used to be called the National Guard, we can't call the resting ones anymore. But the resting T cells are not affected by this. Now, there are, of course, subtypes in the dividing profile too, and that's frankly not completely understood yet, which ones are most sensitive.
But I had the same question that Alan did. Since you discovered this enzyme and pathway in a disease where there's viral susceptibility, you then produce something that's a 4 million selective for that. Aren't you a little bit worried that you're going to reproduce that disease, particularly in terms of viral susceptibility? In that regard, what's happened during the animal toxicologies in this regard?
So what you really don't want to do with this kind of therapy is recapitulate the human genetic disease, because if you do, then you'll have viral susceptibility. We do know that these infants with their natural background population of T cells have no symptoms for over a year. And that's the period, presumably, that it takes to start depleting those clones that grow up, start dividing in response to antigen challenge. So the idea is that almost all T cells are clonal. In other words, it's one clone that loses its growth control, causes cancer. So that will be growing out over the background. You treat that for a period of a month or 2 months, whatever it takes, to kill that clone, and then you should be able to go into remission for a period when there's another clonal event that brings up another population and treat again. So this kind of therapy probably won't ever be good for continuous treatment, but it will be good, we hope, for episodic disease, like most cancers, and, well, T-cell cancers, and also autoimmune diseases, which also have episodic cycles.
Vic?
This is a question for Bill. I missed the first part of your talk. I was at our intern recruitment session. We're trying to create that continuum of, clinical investigators, and we have to do that in the mornings over at the hospital. But I heard your talk before, and I know the Vannevar Bush piece at the start. Certainly the new paradigm you talk about, where going from the patient and the genetics and identifying previously unthought through pathways is going to be extremely important. But I'm just repeatedly struck by the role of serendipity. Vern used the term, the mess, the scratching, in the dirt, the prepared mind. I mean, he held up an aspirin. If you started with all the knowledge of the pathways of prostanoids, you would never, ever pick the drug aspirin, because you would think you would interfere with so many pathways, and yet fortuitously it knocks out platelets because they're terminal, they can't make the cyclooxygenase, and everybody else remakes it and you're fine. So how do you fit this role of just the sort of undercurrent of scratching around and looking for opportunity into the new paradigm? Where is that?
That's a great question, Vic. I think, first of all, the one thing that should not be taken away from my comments is that there should be a lessening of basic research, because basic research and inquiry for its own sake has led to tremendous serendipity opportunities. It sets the stage for serendipity, where somebody's studying an enzyme pathway that thinks nothing that's related to anything, and in fact, it turns up to be a cure for disease. So you can't reduce the investment in basic research. It's still generated for everything else. I think the second thing is how to capitalize on those things a little more rapidly is what you have to sort of consider that. And I think that there's no question that having people come together and talk about what they think is unrelated research and hear things like this, I mean, it almost makes me believe I could understand immunology. Almost. I'm always put in mind of Francis Collins' early days when he was localizing genes and had to go into the database to find out what the gene was they found the mutation on. He said, every scientist I ever know uttered a silent prayer while they were waiting for the result of what they were going to work on for the next 5 years. Please, God, don't let it be an immunologic group. But having said that, it was very clear. I had this illusion that I understood it. But those kind of things where you put people together like this and think about this in a way where this presentation and that presentation cross-fertilize, I think is what we haven't done enough of. And chance favors the prepared mind. And I think having the prepared minds come together and talk about it, you suddenly see things that you would have never seen before. We spend too much time in our own silos. And I think that's the opportunity of the CPSA, is to get people together with apparently unrelated, because smart people make associations in ways that that's how they got to where they are, by being creative and opportunistic in the right way. We just got to create the stage and make a catalytic environment for that to happen. I think departments of medicine are one of those places where it happens. You add the bedside component to it, and if you've got the right teachers, then that's the fodder on which a lot of this grows, I think. So a lot of this is stage setting like today, and Harry put a lot of things together that are a classic example of how you do that.
Nir?
Teresa, that's for you, and it's a scientific question, but related to bedside, okay? So, do you do anything with newly diagnosed type 1 diabetic patients, or can you do anything with their T cells or even their plasma in order to go back and show that your antigenic discoveries are relevant to those patients?
Right, so I mentioned that we have a protocol where we go to Children's Hospital at Montefiore and we get blood samples mostly from new onset kids. And so the studies that I described that Irene had done where she looked at blood samples and asked whether they recognized IGRP peptides, those were all done with new onset Einstein patients.
Alan.
One of the important implications of our investment in NIH dollars is to maximize what we get out of large clinical trials. Even if the result of a clinical trial is, quote, negative, although hopefully if the science is well done, you'll learn something from that, there are resources that should come that bear on your question. So the NIDDK, before my time, had committed to a prevention trial in type 1 diabetes called PPT1. And it really was quite expensive, ongoing, tested the rationale that actually insulin administered both orally and then separately parenterally could tolerize individuals at high risk, and they were defined in a very particular way based on HLA type, family history, and a variety of other things. The New England Journal paper describing the main results of the trial were negative. There was no evidence of protection. But having said that, tremendous amount of resources and samples collected along the way so that one should be able to go back and really get at some of the questions you're asking. But here also, I think it's relevant to the talks we've heard. A lot of the real advances depend on technological breakthroughs. And as Teresa implied, the difficulty, one of the major difficulties in getting at this disease mechanistically is that the T cells involved are not readily available in peripheral blood or plasma. You have to go to the pancreas, which is really inaccessible. But Bill mentioned imaging. So a major investment that we had made, and it's come to some limited fruition, is non-invasive imaging of beta cell mass at a functional level. And there are some advancements there that would be very helpful. And also, I think Diane Mathis at Jocelyn has pioneered this, non-invasive imaging of inflammation in islets. And this goes in the category, I guess, of biomarkers, et cetera. Ways, if Teresa's intervention turns out potentially to be testable in humans, you have a non-invasive way of very sensitively monitoring. So I think this is kind of all in keeping with Bill's very visionary presentation. The opportunities are extraordinary, and this is why it's so important that we don't flag in our opportunities to fund this work despite the financial meltdown.
Alan beat me to the punch. As participants in DPT1, I know that there were samples of cells collected and banked for future study. A couple more questions. Howard?
I don't think I need a microphone.
But you'll get one anyway.
Carrying on with the questions regarding the immune modulation in terms of type 1 diabetes and its clinical relevance, I'm wondering, are there, and cross-pollination between sciences, I'm wondering, have there been clinical epidemiologic studies of allelotypes associated, HLA allelotypes, type 1 allelotypes associated with risk of type 1 diabetes? And if so, would it be more likely that you would find the relevant modulating factors for treating the disease in humans if you were using the animal models produced for those allelotypes?
Okay, so we've known for a long time that the primary susceptibility is the class 2 MHC molecules that the patient expresses or even that the mouse expresses. But now with more careful study, we know that you can separate the effect of class 2 and you can show that there are indeed class 1 alleles that predispose to disease. And the one that we're primarily studying, HLA-A2, is one that predisposes to disease. I have a collaborator that's making A24, which also predisposes. But it's really not clear if you just want to look at those HLAs. We're actually looking at some other HLAs as well, and you do find CD8 T cells that recognize beta cell peptides in the context of other alleles. So those T cell responses could potentially be pathogenic ones, or they could also be ultimately biomarkers for autoimmunity. So I'm actually struggling with this myself right now and thinking of the priorities of my own lab, because it's not a huge lab and mice are expensive. But right now we're studying both predisposing alleles and alleles that appear to be neutral.
So on that note, which Teresa is saying she's looking for collaborators, let's stop for a break.